Welcome back as we continue our look into the digestive system. And so we just left the esophagus and made our way into the stomach. And so in the stomach is going to essentially be kind of a holding chamber for the stomach itself. And so this is where we're going to kind of store the meal as it starts to get slowly broken down. As we can see in this picture of a dissected stomach, the stomach, if you remember back to anatomy, has all kinds of little gastric folds in it, or rugae. And so that allows for the stomach to start to expand and increase its volume inside as we fill it up with our meal that's there. Once it's inside the food, we can kind of take that whole meal and store it in there. And then over a period of time, it's going to be slowly released into the small intestines. And so we're not going to allow it to instantly just go from the stomach into the small intestines. It's going to be a slow, kind of prolonged process of getting it to empty into the small intestines. And so this is going to make it so that in terms of like blood sugar levels, our blood sugar levels don't just rise up real quickly and then come back down. We can kind of rather have a process where the levels are going to slowly rise over time. It's going to give us energy over that period of time and essentially give us that energy that we need all the way until the next meal comes around. Depending upon what's in the meal is also going to dictate how quickly or how slowly the stomach is going to empty as well. And so depending upon what's inside there, depending upon the quality of the food in a sense, is it more liquefied? Has it been blended up and mixed up initially? Or is it in larger chunks, things that it needs to be broken down? In addition to that, the content is also going to matter as far as emptying goes as well. And so the higher the lipid content of the stomach, the longer that food is going to stay in there as well, because the small intestines can only deal with so much lipids at any given period of time. And so that's going to alter how quickly we continue to empty from the stomach. Once the food gets into the stomach, we've chewed the food, we've mixed it with some saliva. So we've done some mechanical digestion as well as a little bit of chemical digestion from the saliva. But the majority of the breakdown is going to happen here in the stomach. And so this is a good proponent for chewing your food well, because the more chewed the food is, the less work in a sense that your stomach has to do with the food once it gets there. But it has several things to help it out in the breakdown of the food. And so the stomach is going to mechanically break down food. And so it's going to break up that food that's there. And so it's going to use its peristaltic waves, its mixing, to bring that food into submission in terms of its overall content that's there. But it's also going to have some helpers. And so one of those helpers is hydrochloric acid. And hydrochloric acid is going to help to kind of dissolve the food a little bit more. It's going to help to break up the food. But at the same time, it's going to help with, since our food hasn't always, in a sense, traditionally been very safe, there's the potential for things like bacteria to be on the food. That hydrochloric acid is going to hopefully be able to destroy any bacteria that came in with the food itself. And so if we store food for any period of time, there's always the risk that bacteria may start to grow on it, and that could also then cause us some digestive problems along the way. And the majority of all bacteria can then be destroyed by that hydrochloric acid. There are some that have the ability to live in there, one being E. coli. And so E. coli has the ability to survive inside the, or not E. coli, H. pylori. Has the ability to survive inside the stomach, and this is what can lead to the production of ulcers inside the stomach as well. So that's going to start to break things down a little bit and kind of just pull the food apart, almost like a marinade does. And so if you have a high acid substance that you're marinating food with, that's going to help to break down some of the textures that are with it. In addition, it's also going to activate another enzyme called pepsinogen. And so we have the inactive enzyme pepsinogen, but in the presence of hydrochloric acid, it then becomes an active enzyme called pepsin. And that pepsin can then now start to digest proteins. And so we're going to start some protein digestion inside the stomach here. Other things inside there for lubrication and general protection of the stomach itself, we have mucus. And so that mucus production here is going to be protective to the lining of the stomach. It's going to keep the hydrochloric acid, in a sense, buffered away from the wall of the stomach itself. And then it adds to the fluid content, making it so that our food has more and more of a fluid content to it, thereby making it easier to mix around. And then we also have intrinsic factor. Intrinsic factor is an important substance when it comes to vitamin B12 absorption later on down the line. And so without intrinsic factor, our ability to absorb B12 is going to decline. And this oftentimes happens with age. And so intrinsic factors levels start to fall as we age, which leads to the potential for a vitamin B12 deficiency, which then leads to the potential for pernicious anemia. And so we want to make sure that we have enough of that intrinsic factor to be able to absorb all the B12 that we need. In the small intestines have the job, in a sense, of taking all of that food that's been broken down in the stomach and getting it into the body. Remember that the digestive tract, anything that's, in a sense, traveling through the digestive tract is not technically in the body yet. And so it's not truly inside the body until we can actually absorb it. And so as food leaves the stomach and comes into the first portion of the small intestines here, the duodenum, it's going to, in a sense, be mixed with secretions that are coming from the pancreas, as well as potentially from the gallbladder. And so depending upon what is in that meal, if there's lipids in that meal, we're going to get gallbladder secretions, we're going to get some bile coming down into the small intestines. And then we're going to get other enzymes for digestion, the majority of our enzymes coming from the pancreas. And so a large number of those enzymes being secreted by the pancreas, as well as from the gallbladder, and that's going to allow us to digest our foods. For the gallbladder, those secretions that are coming from there are geared towards the emulsification of fats. And so emulsification is the ability to take a mixture of water and oil, and normally when we take that water and oil and mix it all up, eventually it's going to settle out into, once again, a layer of water and a layer of oil. But if we put an emulsifier in here and put a couple of drops of, say, soap in there, a detergent, that emulsifier that's inside there is going to start to break up those lipids that are inside. And so now when we go and we didn't draw my beaker as well there, and we go and we break it or mix it up, we now have the emulsifier inside there. We have our water inside there. We're going to mix it all up here. And we're going to have this combination now of, I'll put a little bit of the emulsifier in there with that, and that's going to help to bind them together. It's going to allow us to mix oils and waters together because normally they don't mix. And so the oil is a hydrophobic component, the water being a hydrophilic component there, and normally they don't mix together. But if we throw an emulsifier in there, now we can have, you go to the grocery store and you purchase a, say, an Italian dressing or something like that, and it doesn't really look like you have to shake it at all. That's because they've added an emulsifier to it. It's allowed them to essentially blend it all up and keep all of the components mixed together rather than allowing them to separate out. If it's just simply, say, oil and vinegar, that oil and vinegar is going to always separate out. And so leave it sit there for a little while, and it'll always separate down into the water component as well as the oil component. So in addition to things like our bile salts that are there, and the bile salts that are there are going to be our emulsifiers of our fat. And essentially what's happening in there is it's creating a little bit of almost like a bubble around them. And so that surrounds that lipid. And so in the picture here, we have our lipid at the center. And then we put the bile salts around it. And now that bile salt, since I still have purple here, our lipid in the center there, the bile salt is hydrophilic. It likes water. So it can be around the water, and it's basically just mixed with the water itself. And so it's mixed in with it rather than separating the two out. And so this makes it into a process where we can have them coexist in a sense together, and they're going to be able to be transported through the digestive membrane and allow them to be absorbed into the body. Other enzymes that are going to be released, we have essentially all of our enzymes for digestion coming from the pancreas itself. This is going to allow us to break down proteins, break down carbohydrates. We started the protein digestion in the stomach with our pepsin, but we're going to continue this process down here in the small intestines as well. And so we have protein enzymes, we have carbohydrate enzymes, and we have further enzymes for lipids as well. From there, we're also going to have to be able to, especially in the duodenum, we just came from the stomach, and the stomach has a very low pH. And so it has a pH of around 2. And if we just dump that pH 2 chyme into the small intestines, it's going to burn the duodenum. And so it's going to cause damage to the duodenum. So we need to mix it with an alkaline solution, put it with some bicarbonate so that we can raise that pH up. So we can get that pH back up around 7 and bring it into a more neutral standpoint at that point from there. And then we can continue with that digestion chemically within the small intestine. We're going to do some segmentation to mix those foods around, and they're going to combine together, and that allows us to mix all of the pancreatic enzymes, all of the bile from the liver, all of those enzymes, and get it well mixed around, get it well broken down into the small intestines so that it is then ready for absorption. And so the small intestines is designed really for absorption. And so it's going to add all of the enzymes into it, but we need to have a situation where we have a very large surface area. So we have a large surface area, and that comes from essentially, rather than having the inside of the small intestines being like a pipe or something where it's nice and smooth inside, the inside of the small intestines has folds in it. And then on those folds, we then have little villi that stick up. And we'll look at a picture on the next slide of this as well. And then even on those little villi, we then have, and I can't draw them that small, we then have little microvilli. So we're constantly increasing the surface area of the small intestines, making it so that we have just a huge amount of surface area in order to be able to absorb all of those nutrients that we just broke down. There's no point in breaking down all those nutrients if we can't absorb them. So chemical digestion continues on, and we have now the food ready to be absorbed by these small little cells. And so inside the small intestines, we have these circular folds. On those circular folds, we have these almost like little finger-like looking structures called the villi. And all those little fingers kind of stick up out of the small intestines. And then on the villi, we have individual little columnar cells, and those little columnar cells then have little microvilli. They almost look like cilia on the top of the cells, but they don't move. They're just simply there to increase the amount of surface area that we have available for digestion. And as the food moves through the small intestines, and by this point, to really call it food anymore would be a kind of a misnomer, so to speak. But the food has to come in contact with the walls themselves. If some sort of piece of food were able to kind of travel through the digestive tract, right through the center, and never actually come in contact with the wall, it's never going to get absorbed. So it has to actually get absorbed through the wall of the intestines by coming in contact with the intestines themselves. And so it has to make that contact there. And that's where all of that segmentation and peristalsis is allowing for that ability to mix things up and get it all kind of mashed up against the walls of the small intestines themselves. Digestion itself, in terms of our movement through the digestive tract itself, is controlled by, and we talked about this before, both a myogenic and an involuntary muscle contraction. And so we have muscle contraction being controlled within the small intestines themselves and the smooth muscle that is there, as well as a parasympathetic control over the intestines that are there. We can modify it by a nervous input to the small intestine. So if we're in a fight or flight stage, we can slow down digestion and inhibit that motility of the small intestines. We can increase it with a parasympathetic innervation that's there as well. And then we also have a series of hormones that exert control over the digestive tract as well. And so these hormones themselves are coming both locally within the digestive tract as well as from far away outside of the digestive tract itself. And so these hormones allow us to control a little bit of motility. They control things like our hunger in the first place, our desire to eat within the context of our normal kind of eating processes. So the digestive tract is a very large innervation. So we have our myenteric nervous system that's there. And so this nervous control that's there lies within the walls of the digestive tract itself, basically running through the entire tract that's there. And it's going to influence the movement of our smooth muscles, so our circular layer and our longitudinal layers in there, as well as the secretion through the mucosa and the submucosa of enzymes, of mucus, things like that into the digestive tract itself, so that we have a process where we're secreting substances into the digestive tract based upon the needs of kind of fluid contents that's there as well. And this is constantly looking at what's going on inside the digestive tract, so that as we need to modify either motor control because our food has become mechanically digested well or broken down very well, we can then increase the movement onto the next segment of the digestive tract that's there. This can then, as I said before, extrinsic nerves can play a role as well. And so we can have parasympathetic and sympathetic innervation bring about a change in the motility of the digestive tract as well. So sympathetic and parasympathetic activity runs the processes here as well. And so sympathetic nervous system is going to slow down motility. And so again, this is our fight or flight. So we're going to decrease digestion, versus parasympathetic activity is going to increase motility. And so this is our rest and digest for the system themselves. And so they use slightly different, as we looked at when we looked at the nervous system, they use different neurotransmitters and then different receptors for the process itself as well. And so sympathetics utilizing norepinephrine, parasympathetics using acetylcholine with a muscarinic receptor, and then an alpha-2 nicotinic receptor in the sympathetics there. And that's either going to speed up or slow down digestion, move it through the system or slow it down. Once we've left the small intestines, essentially almost all of our nutrients have been absorbed. And so we're going to have a very minimal amount of absorption needed at this point. Essentially, the large intestines is all about making fecal matter. And so this is making the last of the what's left into something that we can discard, we can do our elimination with, and not lose a large amount of salts and water that we pumped into the system. And so in terms of water, anywhere between 1 to 2 liters a day is put in there in terms of intestinal fluid. Add into that saliva, add into that the stomach secretions, and we have a huge amount of water. Anybody who's experienced digestive upset where you've had diarrhea, the amount of water that is being lost can be extensive to the point where we end up with dehydration. So the large intestine's job is to reclaim all of that salt and water that was brought into the system and get the majority of it back. The large intestines has a large residential bacteria. And so we have a large amount of bacteria there, some of which is going to aid in further digestion. And so some of the things that we consume, especially in the fruits and vegetables category, we're not able to digest. And so we talked about cellulose before as being one thing that we can't digest. And the bacteria that we have don't really help with that either. But there are other substances inside fruits and vegetables where the bacteria that's there allows for the extraction of vitamins and minerals. And so that we can break those vitamins and minerals out of the food. They digest the bacteria, utilize other sugars that are inside the foods that are there, and we're able to absorb those vitamins and minerals that we otherwise would not be able to get access to. We don't have the ability to break them down. So we have to be able to get them from those bacteria that's there. So bacteria are an important aspect of our normal gut flora. And so this is what's normally living inside the gut. It starts all the way from the mouth. We have bacteria that are there, goes down into the esophagus and into the stomach. As we described before, the majority of the stomach bacteria is going to be destroyed, but some of it does make it beyond the stomach and into the small and then large intestines, with the large majority of them being here in the large intestines. Not only are they good for liberating vitamins and minerals, but they're also good for immunity. So those bacteria have been implicated in disorders of the potential immune system, everything from skin disorders to general immunity itself, and increasing our ability to fend off other potential organisms that are there. And so that bacteria is a good thing for us to have there as it helps out with our overall protection. So as the food moves through the large intestines, we started out in the cecum, and we start to move through the ascending colon, transverse colon, descending colon, and so on. The food that was there is now everything that we can't digest. So it's the undigested food that's there. There's going to be some bacteria. Those bacteria generate all kinds of gases as they are breaking down the sugars that are there as well. We're going to have a little bit of water in the fecal matter. We're going to have some of our salts that were left from the gallbladder. And so we're going to have a little bit of bilirubin there in terms of the bilirubin that's there. We may even have part of that undigested food that's there would be things like our cellulose. We're going to have fiber. Some of that absorbable fiber is going to absorb things like cholesterol. So some of that fiber that's in there in our food is going to help to absorb some of the cholesterol that came with the bile salts from the gallbladder. and that's going to help to eliminate. And so this is why certain products in the grocery store you find as being heart smart or helping with cholesterol, things like that. And that's because they have a high fiber content to them. And that fiber is then able to kind of soak up some of that cholesterol in the digestive tract and carry it away out of the body. That bacteria produces a relatively large amount of intestinal gas as they're absorbing the nutrients from the digestive tract there. The majority of that intestinal gas is actually reabsorbed by the body, but some of it does get out, and when we have that, we have a flatus, and so we create flatulence as that does pass. And so this is where control of the sphincters in terms of the anal sphincters and allowing only the gases to escape rather than the fecal matter at the same time. And so with that, we end our look at the digestive system, and we'll move on to the nutritional side of things in our next video.